During Krebs cycle, acetyl coenzyme A will undergo complete oxidation. Since there are two molecules of acetyl coenzyme A produced during link reaction for each glucose molecule, this means that oxidation of acetyl coenzyme A in Krebs cycle will occur twice, which will produce two molecules of ATP via substrate level phosphorylation, 6 molecules of NADH, and 2 molecules of FADH2. At the same time, carbon will be removed as carbon dioxide via a process called oxidative decarboxylation, similar to the process which occurred during link reaction. Krebs cycle takes place inside the matrix of the mitochondria, and the process starts when one molecule of acetyl coenzyme A attached to a molecule of oxaloacetate and produce a 6-carbon molecule called citrate. Next, citrate will undergo isomerization to form isocitrate. Isocitrate will undergo oxidative decarboxylation to produce alpha-ketoglutarate. Oxidation of isocitrate removes electron, which will be transferred to NAD+, and reduces NAD+ into NADH plus H+. At the same time, decarboxylation will remove carbon in the form of carbon dioxide. This is why alpha-ketoglutarate only have 5 carbon. Next, alpha-ketoglutarate will also undergo oxidative decarboxylation to form succinyl coenzyme A, which will also remove one carbon atom as a carbon dioxide and reduces NAD+ into NADH and H+. Succinyl coenzyme A now have 4 carbon atoms. Since we start Krebs cycle with oxaloacetate that have 4 carbon atoms, and now after 2 times oxidative decarboxylation, we end up with succinyl coenzyme A which also have 4 carbon atoms. This means that the rest of Krebs cycle, there should not be any decarboxylation process for the rest of the Krebs cycle. In the next step, succinyl coenzyme A will undergo substrate level phosphorylation to produce succinate, during which a phosphate group will be transferred to guanosine diphosphate to form guanosine triphosphate. After that, GTP will transfer the phosphate group to an ADP to form an ATP. Next, succinate will be oxidized into fumarate. At the same time, FAD will be reduced into FADH2. During this step, the oxidation process is not followed by decarboxylation process, and it's the only step during Krebs cycle which the electron acceptor molecule is FAD and not NAD+. Fumarate will undergo dehydration reaction to form malate, and finally malate will become oxidized into oxaloacetate, which simultaneously reduces NAD+ into NADH plus H+. Regeneration of oxaloacetate means that it can be reused for the next round of Krebs cycle. This is also the reason why the process of Krebs cycle is known as a cycle, since oxaloacetate that was initially used will be eventually reproduced. In terms of the products of Krebs cycle, for each round of Krebs cycle, which means for oxidation of one molecule of acetyl coenzyme A, 1 ATP is produced via substrate level phosphorylation, 1 molecule of FADH2, 3 molecules of NADH plus H+, and 2 molecules of carbon dioxide. However, since there were 2 molecules of acetyl coenzyme A for each glucose molecule, so Krebs cycle will occur twice and produce a total of 2 ATP, 2 FADH2, 6 NADH plus H+, and 4 CO2 molecules.